Thank you, Mary. Now I'd like to introduce Dr. Titia de Lange, who is Leon Hess Professor here at Rockefeller University. Titia is representing the Pearl Meister Greengard Selection Committee this evening, and she will tell us a little bit more about the work of our 3 prize recipients. They're being honored for their discoveries about telomeres, the very ends of the chromosomes. And I should note that Titia herself has made major contributions to this, to the field of telomere biology. Titia has received numerous honors for her research, including the prestigious Director's Pioneer Award from the National Institute of Health, and in 2006 she was selected to the National Academy of Sciences. It's a pleasure to welcome Titia.
Honored guests, distinguished laureates, dear Paul and dear Ursula, in a few minutes, the Greengard Award of this year will be given to Liz Blackburn, Greider and Vicki Lundblad for their discovery of telomerase and its role in the maintenance of telomeres. And it's my task right now to explain what that actually means, how they made their discovery, and why this discovery is so important and deserves this prestigious award. This terrific trio here has brought us insights into one of the most interesting problems in biology, immortality. They found an enzyme that is widely viewed as the key to immortality, and their discovery had wide implications for the understanding and treatment of human disease, including certain aging syndromes and cancer. But they did not start with a quest to cure human disease, or to help its diagnosis, or any other objective that was glamorous and easy to sell to funding agencies. Rather, all 3 of them started with a burning interest into the biology of some pretty humble and unglamorous organisms. Vicki Lundblad made her seminal discoveries in baker's yeast and has dedicated her whole scientific career to this fungus. Liz Blackburn and Greider focus all their intellectual might on ciliates, a single-celled organism that lives in ponds, swimming around, flapping itself forward with long, hairy extensions. You may think you do not know ciliates, but you do. You just know them as pond scum. Imagine this, 3 scientists with a deep devotion to fungus and pond scum discovering the source of immortality, telomerase. To explain what telomerase is, or to begin to explain this, I have to remind you that our genes are organized on chromosomes, which are enormously long strands of DNA that are wrapped up inside our cells. The ends of these DNA strands, the ends of our chromosomes, contain elements that protect the DNA and thereby protect the genes these DNAs carry. And this protective element is called the telomere, for the Greek word for end, telos. Without telomeres, cells can't divide and die because of a host of disastrous reactions that take place at chromosome ends. In Act 1 of this story, Liz Blackburn, when she was a postdoctoral fellow, happened upon the DNA that makes up telomeres in her beloved ciliates. This DNA was very unusual because it was a repeated sequence, many copies in a row, more or less like a genetic stutter. Was this the key feature of telomeres, this stutter? Or was it some oddity of the ciliates, of pond scum? The answer to this question came from work Liz did in her own lab at UC Berkeley, which she had just set up, and then she started a collaboration with a yeast geneticist, Jack Szostak, at Harvard, and together they showed that these repeat sequences indeed are the essence of the telomere. Now, this discovery was a key discovery in itself and probably would have merited some recognition. But it also raised an immediate and very important question. And the question was, where did this DNA come from? Jack and Liz came up with a very bold hypothesis, proposing that cells have an enzyme that's specifically dedicated to the task of synthesizing this genetic stutter onto chromosome ends, thereby ensuring the protection of the genes. And they each went their own way to look for the enzyme. They knew it wouldn't be easy. It was like searching for the proverbial needle in a haystack at a time when in biological research there really were no high throughput mechanisms to shift through hay. So why bother? Why go through the trouble of finding this enzyme, which they weren't even sure existed, especially since they were both starting their labs, they were young investigators. Why take such enormous risks? Well, their motivation came in great part from an old prediction made by Jim Watson of double helix fame and by Olevnikov, a Russian scientist. And both had predicted independently that there is major trouble at the ends of DNA. On purely theoretical grounds, they predicted that our cells can't duplicate the ends of DNA, including the ends of our chromosomes, and they predicted that the ends would wither away with cell divisions. Liz and Jack proposed that the telomerase enzyme might be a special trick cells had to deal with this so-called end replication problem. So they set out to find a hypothetical enzyme that would solve a theoretical problem. Does not get any more risky than that. But the payoff would be great, because if they were right, telomerase would allow cells to prevent death by telomere loss. In Act 2 of this play, a young scientist enters the stage, Carol Greider, who joined Liz Blackburn's lab as a graduate student. She had boundless energy, and still has, as far as I'm concerned, and had no fear of the unknown. And so she was willing to plunge into the haystack, not looking for the farmer's son, but looking for telomerase. This was clearly in 1984, Liz and Carol's yes, we can moment. Carol, guided by Liz, decided to try to dig the enzyme up straight out of the ciliates. She ground up large amounts of pond scum and searched in her ciliate soup for an enzyme that could synthesize this typical telomeric stutter that Liz had identified. Many months later, she emerged from the cold room victoriously, although I'm guessing with very cold fingers and a dripping nose, to announce that she had found the enzymatic activity. She had, in fact, found an enzyme that can synthesize the exact telomeric DNA sequence they expected without any help onto little bits of DNA given to the enzyme in a test tube. The enormous significance of the discovery of telomerase emerged from work done by our third awardee, Vicki Lundblad. In Act 3, Vicki Lundblad joined Jack Stastek's lab as a postdoctoral fellow. Around the same time, Carol Greider was sloshing her pond scum soup in the cold room, and like Carol and Liz, Vicki wanted to hunt down telomerase. Vicki and Jack were geneticists, and so they did what these kinds of scientists do. They sit down at their desk. And sitting there, they think very hard. Rather than getting their hands dirty and cold, by thinking hard, they came up with a clever scheme to find the enzyme without spending any time in the cold room at all. They designed a set of genetic tricks that allowed them to find a mutated version of Baker's yeast that can no longer synthesize telomeric DNA. And the experiment worked. And Vicki found a yeast mutant that had lost the ability to make telomeres. The great insight she obtained from this mutant is that this yeast was initially just fine, even though it couldn't synthesize telomeres. But it gradually lost its telomeres and eventually died. Her mutant apparently could not replicate the ends of the chromosomes without the help of telomerase. This was exactly as had been predicted by Watson and Olevnikov. The end replication problem was real, and it clearly killed yeast that lacked telomerase. Vicki then speculated that what was going on in our sickly yeast strain might also be happening in human cells. What if human cells normally did not contain telomerase? Then their telomeres would also disappear. Did this then explain why human cells at first are fine and divide happily, but after a while stop growing and die? A few years later, Carol Greider, with her collaborator Cal Harley, showed that Vicki's speculation was right. Human cells indeed largely lack telomerase and lose their telomeres and eventually die. When telomerase is put back into these cells, as others did, the telomeres stay intact and the cells are immortal. Together, the work of our three awardees meant that the fountain of cellular youth had been discovered. It was springtime in the field. For the first time, the wider scientific community began taking an interest in telomeres and telomerase. And springtime quickly became a very hot summer when it became clear that telomerase is almost always activated in human cancer, thereby giving cancer cells immortal growth characteristics. Telomerase is now one of the most general markers for human cancer and is studied as a target for chemotherapy. After their first seminal discoveries, Liz Blackburn, Carol Greider, and Vicki Lundblad all went on to make further crucial contributions to the field. Liz Blackburn branched out from pond scum to yeast to mammalian cells and studied in each context how telomerase maintains the telomeres. Carol Greider, with her collaborator Rhonda Pino, made mice lacking telomerase and showed that they too, just like Vicki's yeast mutants, succumbed to the loss of their telomeres. And Vicki Lundblad stuck with her beloved fungus and used its powerful genetics to identify the genes encoding telomerase, which led eventually to the identification of the sought-after human enzyme. Liz, Carol, and Vicki, it gives me enormous pleasure to salute you for your great discoveries. And I would like to ask you now to come to the podium to receive the Pearl Meister Greengard Award. And please could Mike Young and Mary Robinson join.